you, Justin.
Sounds good. And we do have the recording going on this. I want to make sure that that's okay, Chris.
That's fine. Mm-hmm.
Okay. If you want to take a look at the edit, what I did on the last talk was there's actually pretty good function to post that straight from Zoom. I just kind of squeeze out the beginning part here and focus it just right on the talk, and the captioning and everything comes up through that. So.
Yep, sounds great. I've gotten over being perfect.
All right. So I see that we've got participants starting to come into the room. We're going to leave this open and let folks join for the next few minutes, and then we'll start with introductions. So make yourself comfortable, grab your drink, and hold tight for a couple minutes. All right, well, it looks like we've got most of our folks here in attendance, so we're going to get things started with introductions. So very quickly, on behalf of the BioMakers student group and the BioMaker space, I'd like to welcome everybody and thank you for joining us today. This is the third talk in our virtual seminar series, which is COVID-19 BioMaking Solutions. We had terrific talks from Professor Sykes and Professor Yaffe, and we're looking forward to the rest of the summer lineup, starting with Professor Love today. All the remaining talks will be at noons. They're going to be on Wednesdays and Thursdays, so you can keep an eye out for the announcements that'll be coming through the various email lists. I've shown on the screen here two of the email lists you can join if you'd like. They'll also be going out through departmental lists. In addition, I also want to direct your attention to there's a new temporary website that we have up for these talks right here. Links on this page will take you to register for attendance to future talks, so you don't have to wait for the email announcements. And you can also view recordings of previous talks at this location as well. It's temporary, it may move at some point in the future, but we'll try to keep this link active and redirect if nothing else in the future. So one quick reminder before we begin for our seminars, we ask that you hold all the questions to the end. Use the raise hand icon that's in the participants tab, and we will enable you to speak to ask your question directly. If you do have any trouble with that function, please use the chat window and we will try to assist you in whatever way you can. And if you can't get the microphone on or whatever, we can read the question for you. So with that, I'd like to hand it off to Professor Paula Hammond to introduce our speaker.
Thank you, Justin. It's a real pleasure to introduce my colleague in chemical engineering, Chris Love. He is the Raymond and Helen St. Laurent Professor of Chemical Engineering. He's a member of the Koch Institute. He's also an associate member of the Broad Institute and of the Reagan Institute for MGH, MIT, and Harvard. Chris actually found his way to chemical engineering through the field of chemistry. He got his bachelor's degree in chemistry from the University of Virginia and his PhD in physical chem at Harvard University under the supervision of our shared former PI, George Whitesides. After he finished his PhD, he actually settled into research in immunology at Harvard Med and worked with Hide Pluv for a year or so, and also worked at the Immune Disease Institute. Now, he has actually been doing some very exciting things in his lab, including single cell analysis, methods that were pioneered in his lab. He's used that single cell analysis as an approach to understand more about disease and also to discover new therapeutics using a kind of patient-centric, data-driven approach. He's also worked with the Shalik Lab to develop a new portable, efficient method for single-cell RNA sequencing known as Sequel. And he's accelerated the development and accessibility of biopharmaceuticals and vaccines for patients globally. Chris is the co-founder of One Site Biotechnologies, Honeycomb Biotechnologies, and Sunflower Therapeutics. He also serves as an advisor to Squeeze or SQZ Biotechnologies, Repligen, Quantum Site, and other companies. And I think he's an excellent example of a true biomaker, and I'm really excited to hear his talk today addressing COVID-19.
Thanks, Paula. Appreciate the introduction. That was very kind. Excited to be here and tell you a little bit about the work that the team has been doing over the last few months as we've kind of settled into this current pandemic and the challenges thereof of thinking about how do we make sure that everyone can receive access to the right kinds of therapeutic interventions or prophylactic interventions here in the context of vaccines. Let's see if I can share my screen. And since we have a bit of a mixed audience, I thought today what I would do is start with a little bit of kind of rationale as to how and why we think about vaccine manufacturing in terms of scalability and accessibility, as well as sort of the design considerations as to how we might approach this. And I'll tell you a little bit about some of the work that we're doing. This is obviously ongoing efforts and is kind of multi-part in a lot of ways. We have a number of different groups that we are collaborating with around the globe. As we go through this process, and our single largest partner in this activity has been the Bill and Melinda Gates Foundation, who is funding much of this specific work. So I don't have to really tell people at this point about SARS-CoV-2. This is the virus that causes the disease COVID-19. If you haven't been to this website, nextdrain.org, I encourage you to go check it out. It's a really nice interactive tool where you can kind of look at how the pandemic has unfolded. There's a snapshot on the left from late January when a lot of the kind of shutdowns started to emerge in different parts of the world, and a snapshot that I just took yesterday of the current landscape of genomic variants. And I think, obviously there's a lot of challenges in thinking about how do we contain and control this virus and this pandemic in the current environment. And obviously a lot of attention has been spent on thinking about the suitability of vaccines. And I think one nice element for how we think about the utility of a vaccine for a disease like coronavirus is shown in the graph at the bottom. Here, this is a diversity index. So each of the kind of points represents places where there is high variance in the specific locations within the virus itself. And I probably should have thrown one on here for another disease, but the fact that there's only a couple of peaks on here is actually a really good indicator that, in fact, we should be able to look at a vaccine for this disease, and in fact is, I think, quite encouraging about the number of efforts that are going on globally right now to reach that objective. For something like HIV or even annual influenza, there would be many more peaks in these types of plots showing a lot more variation across the disease. So this is, I think, a good indicator for our ability to think about developing a vaccine. I thought I'd start with just a little bit of the immunology, just at a high level, as to how a vaccine works, so that we make sure we're all on the same page as we think about this. The first step in really how having an immune response is an encounter. And this is simplified a bit, but the antigen, the element of the vaccine that mimics the virus in some way, has to be injected and then encountered by the immune system, usually in the periphery, and is in a way transmitted then to other parts of the immune system in the lymph node. So the encounter involves the interaction with something called an antigen presenting cell that it recognizes and pulls down some of these materials, samples it, and then brings it back to the lymph node. It's the informant that now tells a set of cells in the lymph nodes, typically, and other lymphatic tissues, how to recognize, finds the right match for those antigenic components. In this case, it is a B cell population, also requires T cell help in order to fully elicit a robust and protective immune response. But for our purposes today, all vaccines that exist and are commercially licensed today require a antibody-based response in order to provide protection to the individual. So these B cells, there's many different ones. This is the adaptive component of the immune system. They see the antigen in a variety of ways. And once the right cognate interaction has been made, the immune system kicks into gear and makes more of that particular cell. It goes through a process of mutational variants in those cells specifically to enhance the activity of that antibody that it produces in order to be able to bind to the virus, and mobilizes this to produce larger quantities of circulating antibodies. And so you may have seen in the news recently a lot of discussions about, if you've had coronavirus, if you've been exposed, do you have antibodies in your blood circulation? Are they protective? Are they lasting? This is the question that is being asked scientifically, as to how and to what extent are these antibodies sufficient for providing protection to the individual through natural infection. For a vaccine, we want these particular antibodies that we elicit to be able to identify the virus when it is seen and then mark it for destruction. And there's a few different ways that that can happen in the immune system, but now you're interfering with that natural response. We want to be able to block the virus from being able to bind to the receptors on the lung cell or other cell types through the native receptors. And so we need to be able to raise a neutralizing activity to this virus. Today, there's lots of different types of vaccines that are in the marketplace, certainly not for COVID yet, but for other diseases. And historically, our first vaccines were the ones for smallpox, and these used live viruses. They used a variant from cows, the cowpox, as a way to mimic the human version of smallpox. This is a live virus-based vaccine. And with the elimination of smallpox globally, we don't use this anymore, but this is really where the founding principles of vaccinology began. And then over the years, we've learned other ways to make vaccines. First, with reducing the virulence of different viruses to be able to provide an attenuated version of it, say chickenpox or measles, to inactivated viruses that you might treat chemically in order to prevent unwanted infection by the original element. So polio vaccine, this is a current effort in developing and using inactivated polio virus as a way to try to eradicate that disease globally. And then what's happened in the last few years is we've kind of collectively as a community learned more about how the immune system operates and sees different kinds of viruses and what's necessary to train and teach the immune system. We're now at a point where we're starting to think about how do we design vaccines by engineering. And there's really kind of two approaches to this that have emerged in the last several years. One is what we would call a subunit vaccine. This is a recombinant protein that is expressed only a portion of the pathogenic agent, in this case, the receptor from the virus surface itself, and then express that and administer it as a full vaccine product with what's called an adjuvant, a chemical moiety that would allow for training the immune system in the appropriate way when it sees that antigen to elicit the kind of responses that we talked about a moment ago. excitingly and in the news and coming up next in the seminar series, I noticed is efforts in developing nucleic acid-based vaccines. So this in principle skips the step of having to make the protein itself and allows the body to read the RNA or DNA that's directly injected as a vaccine, and then translate that into protein within the cells and now teach the immune system. So it takes a step back. And I think one of the elements of this, as we've seen in this current response, and I'm sure we will hear about next week with the Moderna team, is the speed with which they've been able to accomplish that by synthesizing these genes. Now, one of the challenges here, and I think one of the exciting things actually in the context of vaccinology as a whole with this current situation, is that there are no approved vaccines of this type presently. And so the work that's being done by Moderna and Inovio and a number of other companies, BioNTech, to accelerate these types of ideas in the clinic, I think is going to be really great for vaccinology as a whole. One of the challenges that's still faced, and you might see this in the news today is, as you read about these types of vaccines, is the challenges in manufacturing these. And hopefully maybe next week we'll even hear a little bit about that in the next seminar. Subunit vaccines, however, rely on the kind of existing infrastructure to manufacture large volumes of protein that are used in vaccines today, as well as in therapeutics like monoclonal antibodies. And so represent a really important class of molecules as we think about making a vaccine. So I wanted to talk a little bit about, what do we need to do in order to get a vaccine that's going to cover the entire population, that really is the right scale of coverage that we need. And so I think to do so is kind of helpful to think a little bit about what the disease itself looks like, the epidemiology of it. If we have a disease like COVID, we've heard in the news a lot, this number R naught. R naught is how many persons you come into contact with if you're infected, that you have a likelihood to infect. Seasonal flu is kind of about one other person. The number that's being thrown around for SARS is like 2 to 3. And to put that into context, something like measles, the German measles, which has been reemergent over the last few years, has an R naught value of about 12 to 18. So again, the SARS value is still not fully determined and is kind of in process of being understood, but it's in this number. And so that means that you have a likelihood of being able to transmit it to multiple persons, and there's a lot of efforts through social distancing and other strategies to reduce this to less than 1 so that those transmissions are less frequent. There's also a lot of discussion of herd immunity. This is the natural immunity that arises from interactions. And in this case, I've kind of shown as purple individuals who become immune. So because the individual is infected, can't now interact with the other persons that are unexposed, because of just the sheer numbers here, we get some protection as a population group. But the challenge with herd immunity under natural circumstances is that it can wane for a number of different reasons. It might be that the immunity is non-durable, meaning that you might catch coronavirus again because your antibody response wasn't sufficient, or just the nature of the virus is such that it changes over time through seasonal variations. There's a lot of news currently on trying to ask this question: can you be reinfected? I think that's still an open science question. We don't know. But certainly a reason why you would have sort of concerns about waning immunity overall. And then of course, just with our modern lifestyles of thinking about travel, though not in the current circumstances, this is another reason why population groups can change that can impact herd immunity locally. And so what we really want is, right, and I think this is clear to everyone that's looking at this, that ultimately the challenge is in how do we do this and do it efficiently and fast as a community. How do we realize this? And probably not just one vaccine, but actually many vaccines to be able to cover the globe in this way. But now this allows for this kind of herd immunity, but now doing so in a deliberate and targeted manner. The question is how much of a vaccine do we need? And the number of doses that you need to be able to achieve herd-like immunity is something like the population number times the quantity of 1 minus 1 over R naught. And so for COVID, this is going to mean roughly 50 to 70% of the population, if not more, depending on the final value of these R naught numbers, will require vaccination in order to achieve immunity for the population group. So what do we do? What do we go after? What's the strategy that we want to adopt here? We need to come up with a plan of attack on this. SARS is this coronavirus, as we've seen. We've all seen pictures like this now in the news. The spike protein is the piece that's accessible on the outside. And this is the element that we know from other previous coronaviruses is responsible for mediating entry into the cells that they target. So this is a trimeric structure. It sits on the surface of the viral membrane and it interacts with the host cell membrane. The cartoon here is for MERS, another coronavirus that was identified a few years ago and actually is in some ways more virulent in its prior epidemiology studies than the current one, but interacts through a very similar kind of mechanism. Below that cartoon is shown the cartoon of the spike protein itself. So it's the monomer unit. These are all the different elements to it. And so what we'd like to do for a subunit vaccine is to think about, could we make that protein, and do we need to make it as a trimer? Can we use a portion of it? How would we think about the design of that element? But the spike protein itself is very efficient for this vaccine strategy because it is the element that is necessary for the virus to be able to gain entry into the cells. And it so far has shown very low evolution in the spike. There's one site that has been identified that's being associated with a more virulent strain, but overall does not have a large impact on the structure of the spike protein. And so if we develop a vaccine now, it should apply for the variations that we see down the road. This is different than, say, seasonal flu or say HIV that has a very high degree of variance, so that we're always playing catch up in our vaccine design. So globally, we should rest assured and feel confident right now that we can develop a vaccine that will be meaningful in a year's time. So one of the challenges and one of the things that we've been thinking about as a lab over the last several years is it's great to be able to make vaccines. They're important, or prophylactics or other biotherapeutics, but everyone should have access to these in some way. And one of the challenges in doing this is ultimately in thinking through the manufacturing. It doesn't sound as fun as the discovery piece of finding the right molecule or the right antibody, but the manufacturing itself is actually what delivers these therapeutic concepts or interventions like vaccines to a patient. And so one thing that we try to do when we work with groups like the Gates Foundation or some of their partners is to start with the end in mind. We want to think about what is the target product profile. It's not simply the protein itself, but it's actually the protein itself in a formulation, in a vial, intended to be used in a certain environment and in a certain population group. And all of that kind of structures how we think about constraints on the engineering problem now to develop a vaccine efficiently and engineer it to suit its purpose. And so I've kind of outlined here some of the different things that we might consider and think about as we go through this. I won't highlight all of them, but we want to be able to reach a very large population. Ideally, we want a single dose because already we're going to have to reach a very large population. At most, we want 2 shots. People just don't like shots, so that's another element to consider as well. It's going to have to be adjuvanted. What's the right choice of adjuvant? Historically, alum is used. Are there kind of newer agonists and information that we know from the immune system might shape a different choice of adjuvant here in this example that might help facilitate getting to that single dose. But now the protein that I make has to also be compatible with those chemicals in the right way. So I have to formulate it properly. For global use, it needs to be stable for long periods of time and kind of liquid formulation at kind of minimal refrigeration. And the cost should be low. I mean, obviously $1 a dose seems pretty expensive. Maybe in this case, that's a reasonable amount, but 15 cents a dose or less for the final product would obviously be much better to be able to reach all parts of the world. And so, kind of what I've shown here is the type of target product profile that we would develop with like the Gates Foundation or some of their partners to try to really think through the details of what goes into it. But the bottom line is ultimately a vaccine that's of this volume, we're going to need a lot of material. In this case, I've kind of estimated about 500 kilograms. And that's something that we're going to have to now really think through as we go forward into the development of the subunit vaccine. To put that in context, the spike protein that I showed you in its full trimer glory, the whole thing is about 400 kilodaltons. And remember, viruses aren't really made to be produced in large volumes. They kind of hang out. They're looking for ways to kind of survive and move along. They don't need to make very many copies of their particular spike proteins, and some of them just aren't productive in any event. So these are really large proteins. To put it in comparison, something like a monoclonal antibody, we make about, as a community, kilograms to maybe a metric ton of these types of molecules per year. Examples are Herceptin or Keytruda or some of the COVID therapeutics that are in clinical trials now. It's about 150 kilodaltons. As we start to move down in molecular size, insulin, about 6 kilodaltons, we make about 10 billion doses a year and we make about 1,000 kilograms. So this is a very large volume manufacturing, and then you keep going, small molecules. And so that you can see there's this inverse relationship between the amount that we kind of make annually and the size of these proteins. And so for where we like to be able to get to in kind of maybe 100 kilogram-ish order of magnitude, we can start to run some numbers and say, like, look, if we want to be able to get to 500 million doses, we're going to need to look at maybe large reactors, 50,000 liters. We're going to need to be able to get to gram per liter titers. We'll have to purify and recover that, be able to make about 25 kilograms per batch, something like that. So we would like a molecule that's going to be in this kind of size range between about 150 kilodaltons and 6 kilodaltons to be able to kind of accommodate that easily with the existing manufacturing capacity. Try this again. Here we go. Apologies for the interruption. So how do we make a vaccine? We got to start with cells. They have to express a component of the vaccine. We're going to put those into a bioreactor. I mentioned 50,000 liters. This is an enormous reactor. You might be familiar with one in the bench in the lab space. That's a couple of liters that sits there. You want to control all of the elements, pH, oxygen, conductivity, other aspects of the media in order to optimize the productivity of that. We then recover it, usually by chromatography methods and some buffer exchanges and elements. And then we have to put it in the vial with all the other components that make it a product. And so these are the kind of steps that go into it. How long does it take to make these things? Mammalian cells, if you start with that as a production platform, is maybe a month, roughly, by the time you kind of turn everything over in the facility to do it. Microbial systems, in contrast, are about half that time. They grow about twice as fast, roughly, give or take a little bit. And so overall, you can run your facilities faster. In a pandemic, this seems like a really good idea, is to be able to manufacture on a faster time scale. And so ultimately, the number of doses that you get per batch in your manufacturing is going to be dependent on all of these different factors: the choice of your cells, the volumes of production, the scales at which you operate at. And that's all constrained, at least today, by what facilities exist where. We don't, we can't build a facility very fast. You might have even seen in the news Bill Gates talking a bit about the timelines to being able to get to large volume manufacturing facilities. These take usually 3 to 5 years to build and commission for manufacturing. That doesn't align well against the current pandemic. And so we need to be able to figure out, how do we take advantage of existing infrastructure today? Very interesting. Love Zoom. You start thinking about where can we manufacture today and what would it take to get there. This is analysis that Drew Peterman put together from my lab, looking at the overall capacity today and kind of yield curves. And we won't dwell on it for very long, but the point is that along the x-axis is the dose size. For a typical recombinant vaccine, you'd like to be between maybe 10 and 50 micrograms per dose. On the left-hand axis is global capacity for manufacturing. And what we've done is we've looked at what's really like the space-time yield of that capacity. So a number of liters that are available in a bioreactor times the number of days that are available to do that, and goes up to roughly 5 million liter days in that context. And so at the very top end of that scale is the global manufacturing capacity. If we took every reactor on the planet and ran it once, we would be able to make a certain amount of volume of material. And then down at the lower end, you can see an example where if we have a 10,000 liter reactor and we run it for 100 days, that's about 50% of the capacity of a single facility. It would be in that range. And so what you can see again is kind of along this axis and marked with the purple dashed lines here is that kind of yield curve of the size of the products. And so if we want to get to very large volume without using too much of our global capacity just because of availability or otherwise, we need smaller products that are going to be high volume products, so kind of gram per liter titers. And so this gives us engineering targets for how we think about where we want to be in the production volumes. All right, so what does that mean for us? How do we think about this and what are we actually going to focus on for our vaccine development now? On the left-hand side is the trimer. You can see this little green region at the top. This is called the receptor binding domain. And normally it's kind of shielded and hidden on the virus. And then kind of as it's kind of getting close to the receptor, it kind of unfolds a little bit and peeks out. Cool thing about this RBD construct is it's pretty small. It's about 20 to 25 kilodalton, so it meets that criterion that we talked about before. It's known to be able to elicit SARS neutralizing monoclonal antibodies. The example that's shown here is this one from previous SARS infections of the old 2002 virus, CR3022. Number of papers that are starting to show up in the literature and a bioarchive that are mapping out the human responses to the natural coronavirus now. And there are many, many neutralizing antibodies being identified that bind to the RBD. So this is good validation that this target, if we use it as an immunogen in a vaccine, should be able to elicit a neutralizing activity. It's also the element that binds to ACE2. So if we develop the right antibodies to it, we're achieving that goal of being able to block interaction with the ACE2 binding site. And because it's small, there's a sort of immunological context for those that are aficionados that it sort of should reduce any concerns, safety concerns around antibody dependent enhancements. So for low cost, high volume vaccines, this is crazy. This is a really exciting target to be able to think about. So some of the work we've done over the last several years in a program that's called Ultra, this is a grand challenge that we've been working on with the Gates Foundation in collaboration with Kansas University and the University of College London, has been really thinking about what if you could design for manufacturability to achieve cost targets and volumes early in the development. So instead of discovery, kind of coming up with a cool idea and saying, hey, guys in manufacturing, figure out how to make this, could we go a step back and say, okay, we want to build all that cool discovery stuff in. It's got to do the right things. It's got to be immunogenic and safe and efficacious, but it also has to be manufacturable. That thinking has not historically been done in vaccine development. And so what we've been working through is kind of how do we start with something like this target product profile and turn the knobs that we have as bioengineers in the molecular design itself, the genome of the host that's making that product. And then ultimately, how do we implement these things on efficient manufacturing platforms, chromatography, fermentation, all of the elements that go into that to be able to really intensify these processes. And so this example that's shown here is sort of around a non-replicating rotavirus example that we had undertaken with the Gates Foundation to take what was an E. coli product and to figure out how do we really intensify that manufacturing using a yeast-based organism. And so through iterative designs of minor sequence changes and some modifications we made to the host, we've been able to push up to kind of gram per liter or better titers and productivity of some of these antigens as a case study. And so when COVID has come along, they called us up and said, hey, can you guys help us start thinking about doing this again, but now for this new situation. And so Sergio in the lab took the sequence for the RBD, which is shown over here on the left, and ran it through a bunch of different tools that we use to assess kind of the qualities of this and kind of take a first pass at manufacturing assessment. How hard or easy is this going to be? And we compared it to some other things that we have experience in making in the lab, like GCSF. And so now you can see kind of these two look pretty similar in some ways. We make GCSF quite efficiently, and actually there's some characteristics of the RBD that are even better. So hey, this looks pretty manufacturable from the beginning. Let's give it a go, see what we can do. This seems like a really good target. And so we can kind of go through our kind of standard workflow of taking that sequence, we can put it into a vector, we can transform it into a yeast, and then we can kind of iterate through this all in the course of just a couple of weeks. And so rather than going through long design cycles that are often the case with mammalian-based systems where you're waiting for the cells to grow up between iterations, and you might have to pick a clone that's suboptimal just simply to move along to representative material for your clinical testing. Here, we're actually directly building into the strains, the genes to allow for the selection. So our clones that we select after this development is the clone that could go on to become the manufacturing strain at a later stage. And so the example shown here again is kind of iterative designs through this type of cycle with the rotavirus that we had previously done, but the same workflow we're now applying to the RBD work. And so when they called us on Valentine's Day and said, hey, here's the sequence that we've gotten now, can you guys like start? What would it take? And so, you know, the team kind of mobilized. This was kind of while the ramp down was going. We got special permission to keep going in the lab. We had a team of 4 that worked, you know, really nonstop over this period of time. But in 29 days, we went from a DNA sequence to fully purified material that was clinical grade that we then took forward into animal studies. The lab went back and we also then did work where we further evaluated how to improve the strains using some of those other levers that we have on trying to understand what were the limitations in the cells to, you know, how do they see the production of it, and kind of in parallel track to our immunogenicity studies, which are kind of next on the development path. We were able to do that. 29 days from start to finish, that was exciting. We had some partners that were interested in being able to take this into manufacturing for clinical studies in India. We started to ship the material to them. It took just as long to get the materials to India in this current environment at that time as it did for us to go through this entire process. So I'll come back to that point a little bit at the end as to where we might go next. The proteins that we've made here themselves, we've put on a platform for manufacturing that we built and developed under some funding from DARPA, the Defense Advanced Research Project Agency within the DoD. It's a fully end-to-end automated manufacturing system. And so we were able to take those strains and put it into this, and again, in a couple of days, be able to generate you know, milligram materials of some of these constructs that we were testing. So that was more than enough to be able to take into our animal studies to advance these concepts. Fully automated, fully end-to-end, very cool. The paper's there if you're interested in reading about it. But you know, the real question is, how's the vaccine going? What's that look like? So the materials that we made in this first round of development, you know, looks great by mass spec. We have, you know, effectively a single peak that's got glycosylation at one particular site. It's hypermonosylated. This is a common glycosylation feature of yeast. In this case, you know, it may actually provide some additional benefit in immunogenicity. If the worst case is just, you know, an extra carry along in this particular context. It binds to human ACE2. It binds to the SARS neutralizing antibodies. And because it's high quality of the kind of manufacturing strategy we talked about, we were able to take all this material into animals, and we're kind of awaiting the results on that from a few different studies looking at different formulations. But so far it looks immunogenic as we would expect. We're also going back and now testing additional variants on this where we've kind of learned some things along the way about other sequence variants that we're exploring presently. From a manufacturing standpoint though, didn't look so great. We weren't able to make a whole lot of material, and the purification yields were actually pretty low through our first round. So we did what all good biology folks might think about doing, or what, you know, what a modern bioengineer might do. Not really a traditional process engineer twiddling knobs on a reactor, but, you know, good bioengineering. We're going to do RNA sequencing. So we did this. We analyzed how did the yeast see the RBD from the COVID that we were making compared to those rotavirus antigens that we were making really, really well. And what's kind of interesting is what comes up is they're really struggling to get it out of the ER. And they said, so there's something going on here. What's the deal? So we've gone back and we've made some changes into the strains. We've made some changes into the sequences. We've changed some ways that we're inserting the protein into the ER, a few different changes that we've been able to make to really kind of take a look at this and enhance overall production. The other thing that we did, because we have multiple levers now, not just at the host level, but also at the reactor level, is we've formulated the media to kind of slow things down a bit. Sometimes you have to go slow to go fast. We were just putting so much pressure on the cells to manufacture this at high volumes that, you know, effectively they were kind of getting stressed out. And so by backing off a little bit with a more simple media feed with a little less methanol, in this particular case, a little more sorbitol. Drew in the lab was able to figure out and demonstrate ways to get to 3 to 5x improvements in the overall production with the new defined media. And so that kind of puts us at a point now where, based on all of the improvements that we've made in the strains and the feeding process, we're at about 0.4 mg per liter per day of the wet cell weight, so normalized by the amount of cells that we have in one of these systems. And if you sort of push that out now into a fed batch titer, that's about 1 gram per liter. So you start to then kind of project that up, and we're kind of in the ballpark of where we'd like to be for these types of processes to be able to manufacture hundreds of millions of doses in small numbers of runs. But of course, you know, we're not really satisfied there. We've been doing some other things, and Drew in the lab has gone back and done some things to modify the yeast even further. And we're getting another boost from that particular set of changes that help with the metabolic loads in the cells to allow for enhanced production of up to about 5-fold in this case. And so now we're starting to really kind of layer on multiple elements of our kind of design cycle here in these approaches. And so, you know, we're super excited because some of these things are starting to move towards clinical development. We're working on 2nd and 3rd generation vaccine concepts now that we think will be kind of even better and more accessible than the first ones that we've gotten out the door. We're doing a lot more immunogenicity and formulation studies currently with several of these different candidates, as well as adjuvants. Excitingly, we've now transferred strains to 2 large volume manufacturers who are advancing these towards clinical development presently. That's been super exciting and super fun, you know, kind of just cool as a lab that we've been able to help with this in a meaningful way now. Our process improvements on the downstream up to where we're at about 50 to 60% efficient in our recovery. And so now we're kind of starting to look like what's a commercial-like process for these things. And that's just kind of the start. I think, Neil, we were looking at the list yesterday. We have about 100 different designs that we're kind of thinking through in variations right now. We need to start down selecting on some of those to start prioritizing again, but I think, you know, just really speaks to what's possible today and the ability to iterate in biology very, very, very fast. Last, we're just right there. Let's see, one more time. All right, let's see if I can share this last bit. There's some lessons learned in what we've done here that I think are really important that we need to think about as a community holistically. And how do we react faster in a pandemic? Every month counts. And I mean, we've been on numerous phone calls now where you can just sense the urgency in the manufacturing group with the partners, with people that are kind of thinking through this deeply. Like, how do we get it there? You know, it was so frustrating when we had gone through all this initial set of work and we then stuck the yeast strain on an airplane in dry ice to get it to India, and then it sat on tarmac. Really frustrating, right? It took like a month to be able to get those strains transited through all the systems because of the various things that were going on around kind of national borders closing and reduction of flights, all for the right reasons. But in this case, you know, really kind of created a barrier and delayed some of the aspects of what we were trying to accomplish there. The other challenge is the processes that I've told you about are ones that transfer well into different kinds of facilities. But the other thing that has to happen now, because we are looking at different kinds of manufacturers for different kinds of molecules, they have to fit it into their facility. And one facility for one vaccine looks very different than the facility for another vaccine. And so everybody has to now do their own development work to figure out, okay, you know, Chris and his team had said, you know, do this and do this, but we actually can't do that. We have to do this other thing. So we're going to now do the experiments to show that those are the same. That takes time. And in the end, it'll allow for scalability, but it's a challenge if we don't have kind of modular facilities and processes. And then I think the other element in all of this too that we've thought about is we don't have the same experience in manufacturing globally with the same organisms. You know, one company likes one strain, one company likes another strain, somebody else uses a whole different organism. All of these for a variety of different reasons that we could talk about for hours. But today with biology and tools like genomic sequencing and CRISPR-Cas9 and gene editing, it's really possible to write biology like software. And I think many of us that are in this kind of space and thinking about BioMaker and what you can do are thinking about how to use those tools efficiently. We need to figure out how as a community to invest in building these kinds of basic software packages on an open source platform for biopharmaceutical manufacturing. And in doing so, build an ecosystem that allows for better interactions and faster transfers. What if I'd been, instead of having to ship cells to someone, I could just ship them the information and they would create the DNA on site somewhere and then generate the strain. And then we could show that our strain and their strain were identical by genomic sequencing. And then they could do a couple of simple experiments to validate that what we see in our media is the same as what they see in their media. That tech transfer process goes really, really quickly and efficiently. So as we start thinking about pandemic readiness, we have to kind of rethink these systems. And I'm hopeful that the kinds of lessons that we're learning here will allow refocusing on manufacturing and kind of the global needs for being able to do so in the kinds of circumstances that we're facing right now. I'm going to stop there. And I think what it would do is be happy to take questions. I'll just quickly thank the folks that were involved in the work. This is the team of 4 that got into the lab right away, and they were all in there working really hard, basically every day for about 3 months. Laura has since graduated and moved on. Drew, Neil, and Sergio are still in there, and now with the ramp up on campus, we have a few more hands around to help as well. And then just numerous collaborators that we're working with through the Gates Foundation network on these particular activities. So thanks for the time, and yeah, certainly be happy to answer any questions. Thanks, guys.
Thanks, Chris. So anybody who has questions, if you'd like to raise the hand in the panelist window, I'll get you called on. Actually, Robert, you had typed one into the chat. Would you like to just ask that out loud?
So the question here is, when do phase 1 trials begin? Will the FDA provide emergency use authorization to begin phase 2 trials? So right now we're looking at trials overseas, a couple of different locations. I probably can't share what that looks like, but the intention is to have those start later this summer or in September-ish is kind of the goals. So we're hoping very soon. I think it also just raises a better point too around what is this going to look like? Everybody is talking about a variety of different products in the news currently. There's a lot of different formulations. I think we need to try them all. I think obviously I kind of projected numbers. We're going to need things along a wide range of cost scales. We're going to need a variety of different products because different people are going to respond differently. Our availability of large volumes of any of these is going to be limited for a period of time here. So I think this is as many shots on goal as makes sense, need to be pressed forward in development. I think one of the things that we're going back now and trying to do is okay, there's going to be this whole wave of things that happens here first, including, hopefully some of the things that we've been involved in already, but there's going to need to be 2nd and 3rd generation candidates too. We're going to have to reach to a point where this is very efficient. It is a single dose, and some of the biology that's starting to emerge is allowing us to kind of go back and rethink the design of the immunogens explicitly to be able to facilitate that with this manufacturing in mind.
Great, Chris. I'm just wondering, is the entire RBD domain being used in the vaccine, or is there a subportion of it which is more immunogenic?
Yeah, so right now, so we have a minimal unit that we had started with based on kind of prior literature and what had been reported for similar kinds of coronaviruses, but we're now exploring a variety of different extensions of that to try to see if we can improve, catch a few more T-cell epitopes and other things.
Super, great work.
Thank you. Yeah, so there's a question here from Helen. What kinds of efforts are underway to develop the kind of cooperativity that's needed to produce a vaccine? I think that's a great question. I think it's one where I'm hopeful that there will be a number of conversations that emerge after we kind of are able to slow down collectively from what's going on presently in this whole circumstance to, as a community, really talk about this. Who are all the right stakeholders to really think deeply about this? We've been involved in a lot of conversations around pandemic readiness through our work with the DoD over the last several years. I know that governments are thinking about this deeply. I think foundations are certainly thinking about this. There's efforts like CEPI that are making specific investments to be able to build capabilities globally. I think, the kinds of things that I was talking about with some of the host cell biology, this is something where it's going to take a team effort, and there's an opportunity to really share risks in that. And actually, stay tuned. There'll be an announcement next week about some work that we're doing there with some of the biopharmaceutical companies and the Gates Foundation to try to establish a framework for how we might think about this type of intentional engineering from first principles about about the host cell biology that's necessary. Today's a totally different era. If we had had the kinds of tools for genomic sequencing and editing that we had 30 years ago when the biotech sector was getting started, we wouldn't have the kinds of hosts that we have today. CHO cells are fantastic. They make many, many products globally, and they will for a very long time. But they're difficult to work with. They take special care and nurturing. Our ability to engineer eukaryotic microorganisms with these kinds of tools really lets us think differently about how we would make a product today with everything that we now know. Let's see, there's a question from Nimrod. Backlog of protein synthesis in the ER. Yeah, so kind of what are we doing to kind of think about disulfide bonds or kind of tinkering with the pathway itself? I think, we've been pretty fortunate in what we've seen so far with the RBD is that a lot of the backup in the ER seems to be more cell-based, that there is an aggregation tendency of the product that we made first out of the gate. And that aggregation tendency is a challenge from a manufacturing standpoint in terms of quality. It's also a challenge that's seen by the cells in terms of how they efficiently process it. You can imagine all that stuff getting clumped up in the ER. It raises its hand and says, well, I'm done. I can't make this efficiently anymore. I'm going to go start breaking down proteins. It's absolutely not what you want to have happen when you're making these things. So Sergio and Neil in the lab are kind of working now through some different sequence variants and potentially, some ways that we can mitigate the host cell biology to overcome that. We have some nice sequence variants now that are starting to be more manufacturable and less aggregation prone that seem to address that particular issue. So, we're hopeful that maybe these next generation candidates will be even better on that front. Question from Virat, do you anticipate long-lasting changes to vaccine development, manufacturing, testing, and collaboration as a result of the pandemic? Yes, I mean, I think in a lot of ways we have not, as a community, spent much time thinking about vaccine development. They have tended in the past to be commodity pricing. There are billion-dollar products in this space now, so pharmaceutical companies are interested in producing them in certain indication areas. For infectious disease, though, this is still a challenge. So we have diseases like tuberculosis and malaria and HIV, and the ones that are kind of in the kind of typical infectious disease category, where we don't have capabilities or current ways to get to that. I think, the idea of like nationalism around manufacturing and kind of just securities and supply chains, we're also seeing kind of play out in this current pandemic. Who's going to get the first lots of vaccine? I think this means that we have to think differently about manufacturing. So as part of the work that we've done with the Gates Foundation over the last couple of years is to try to press into this historical model around centralized manufacturing. Today, most of the vaccines globally are made in very, very few facilities around the entire world, and they supply to everybody. And that works except for when it doesn't work. When you miss a batch, if you miss a lot, you miss immunization targets. So there might be a whole birth cohort that goes for a period of time globally without having access to certain vaccines. And this happens every year right now. I mean, this is not an uncommon case. The reason that they're manufactured like that is an economies of scale question. But the kinds of intensification, the kind of automation, the kinds of things that I showed here, there's a different way to think about regional manufacturing facilities that would provide the kind of capabilities in a pandemic to provide local regions with manufacturing capacity, but would also allow for other kinds of more efficient. And so with cost modeling that we had done with Susie Farid's lab at the University of London, we've been able to demonstrate and show that in fact, we could use a regional manufacturing strategy and meet cost targets that you would typically see from large scale centralized facilities. So we're hopeful that actually as we go forward, and kind of in the community as a whole with the industry and regulators and the government stakeholders to think more deeply about what does this kind of look like and how could we enable this capability for the future, probably for multiple modalities. There's gene therapies and RNA and proteins like we talked about. Cell therapies by nature require kind of regional manufacturing in most cases today. So I think this is something that's kind of coming along, but I'm hopeful this will accelerate it. Any other questions?
So Chris, I had a bit of a technical question when I was looking at the inverse relationship between the size and the amount produced as far as production of various biologics or therapeutic compounds. Is that driven by challenges and difficulty in production associated with the size scale of the compound, or is that something more about the historical paradigm of therapeutic development? And does that have fundamental limitations that are driven by the underlying biology? And what do you think are the biggest factors of that?
Yeah, so I think at a very high level, the relationship makes sense to be inversely related. At the end of the day, the cell is a catalyst and it just takes raw material and converts it into final product. And on a molar basis, a bigger protein is just bigger. It requires more stuff to start with. So just your catalytic efficiency through there is not as good. So you would anticipate the bigger things just on kind of good classical chemical engineering principles of mass balances would generally be less. That said, right, I mean, we don't fully understand the rules of what that relationship looks like in all cases. Some cells are better at making certain things, and certainly post-translational modifications like glycosylation and chaperones for folding, these are important elements that are required to make certain sizes of certain molecules. There was a great question here about disulfide bonds. That's another element that requires the right kind of environment for those disulfide bonds to form, and usually with chaperones. So I think, the challenge is there's lots of different hosts and there's lots of different kinds of proteins. How do you start to kind of build a rule set around that efficiently? And I think, part of what we're trying to do with taking a yeast-based organism is say, look, that's a pretty good scaffold to be able to bring in and layer on top of that enzymes from human chaperone pathways in glycosylation or in folding and things that we know that we can kind of layer in specifically and start to ask, like, can we improve those transfer functions through there? So I would say it's kind of certainly related to protein sequence, also related to host biology, but then also just sort of intrinsically limited by just kind of mass balance considerations. So mapping out all of that, I think is to be more predictive. It is an exciting and interesting opportunity.
Do we have any other questions? All right. Well, I think we can draw to a close then and wrap up. So I'll thank on behalf of everyone, thank Chris for giving us a very exciting and interesting talk today. It's amazing to see the things that can come along in such short time periods.
Yeah, thank you guys. Thanks everyone for joining. Stay well.
And we look forward to seeing everybody next week for our next talk. Have a good afternoon.